Welcome to Keep the Faith Ministry News. I'm Sabrina Peterson, and I'm filling in for Pastor Mayer while he's on medical leave. Scientists from ETH Zurich have announced a major breakthrough and a world first. For the first time anywhere, they've managed to get an artificial intelligence algorithm to create an entire bacterial genome. The findings were published in the Proceedings of the National Academy of Sciences and will have significant implications for the field of synthetic biology. It also now means that Harvard and MIT's goal of creating the world's first artificial human after 2035, a project they've been working on for a couple of years now, is also increasingly feasible. The new genome, named Caulobacter ethensis 2.0, is not a living organism yet, and only exists as a bundle of DNA. Nonetheless, the achievement represents a giant leap towards creating so-called synthetic life and world's first truly synthetic life forms, the likes of which we'll have never seen before. The new genome was derived from that of Caulobacter crescentus, or simply Caulobacter, a naturally occurring bacteria found in spring water, rivers, and lakes around the world. This harmless bacteria is commonly used as a model organism in the lab. Its genome contains 4,000 genes, however, most of this is considered junk DNA, and only around 680 of these genes are actually needed to support the survival of the bacteria in the lab. Moreover, this minimal gene set still contains some redundancies, since several different combinations of amino acids and proteins assembled by DNA often have the same result. So, the scientist in question developed a computer program to determine the ideal DNA sequence, an optimized genome that didn't contain the junk and didn't contain the duplications. The new algorithm managed to completely rewrite the bacteria's genome as new sequences of DNA that didn't resemble the original ones, but that still managed to perform the same biological functions, resulting in the minimal gene set. The latest research builds upon previous work of American genetics pioneer Craig Venter, who was the first to chemically synthesize the genome of a bacterium, although the feat took 10 years to achieve. Whereas Venter and his team made an exact copy of their bacteria's genome, the new computer-generated genome contains an entirely new set of genes generated by the algorithm. Either way, creating a bacterial genome completely from scratch is not an easy task. A step-by-step approach is needed. So, starting with the minimal gene set of Caulobacter, the team at ETH Zurich first created 236 genome segments. Next, the segments were pieced together. While this might sound quite easy, the process is incredibly challenging, since DNA molecules can stick to each other and often become twisted and tangled. Nature also has built-in genetic redundancies, which means that many genes can encode for the same protein. In this case, the researchers rewrote the genome using completely different genetic sequences, and it was still capable of providing the same biological functions. To test out their artificial genes, the authors produced strains of bacteria with both the naturally occurring Caulobacter genome and segments of the new artificial genome. When they switched off the natural genes one by one, they found that 580 of the 680 artificial genes were functional. This suggests there is still some room for improvement before the algorithm can create a fully functional computer-generated genome that can be used to synthesize a living synthetic bacterium. Nevertheless, the scientists hope the algorithm will one day produce functioning synthetic microorganisms for various biomanufacturing applications, like producing pharmaceutical molecules, vitamins, or even vaccines. But not without discussions among scientists and society as a whole on the appropriate uses as well as potential abuses of the technology and how they can be prevented. The antediluvian world was highly advanced in technology, but the people used their great powers only to corrupt themselves. We are fast approaching a similar period in history. Society at the present time is corrupt, as it was in the days of Noah. To the long-lived antediluvian race, only a step from paradise, God gave rich gifts, and they possessed a strength of body and mind of which men now have but a faint idea. But they used his bounties and the strength and skill he gave them for selfish purposes, to minister to unlawful appetites and to gratify pride. They expelled God from their thoughts. They despised his law, trampled his standard of character in the dust. Fundamentals of Christian Education, page 421. This is Keep the Faith Ministry News. Thank you for watching.